Here we go for the laboratory diagnosis of HIV. Diagnosis of HIV/AIDS is not like other infectious diseases. A number of ethical, legal, and psychosomal issues are associated with positive HIV status. So, care should be taken while performing HIV testing. Remember 3 C's when you are doing HIV testing. First C is consent in written format before sample collection. Second is confidentiality of test result is a must, and counseling. Counseling which is pre-test and post-test counseling should be provided. For laboratory diagnosis of HIV, sample is collected, and which sample is collected? It is the blood sample. Blood sample is collected for detection of antigen, antibody, or for detection of viral RNA. There are different types of tests for diagnosis of HIV infection. These are specific and non-specific tests. Specific tests include screening and confirmatory tests. Screening tests are usually used for detection of antibody. These tests should be highly sensitive and specific. Example of a screening test is ELISA. This is the instrument which is used for the ELISA testing. The advantage of this ELISA test is that it can be used for multiple tests, multiple number of samples. The only disadvantage is that it takes around 2 to 3 minutes, 2 to 3 hours. So that is why certain rapid tests are recommended for diagnosis of HIV by screening test. These rapid tests detect HIV and HIV1 and HIV2 either separately or together. ELISA is the most commonly performed screening test at blood banks. They are easy to perform, large number of samples can be tested, and it is sensitive as well as a specific test. This is ELISA washer. Then we will come to the rapid or simple test. These rapid or simple tests are carried out within 30 minutes, or the result is obtained within 30 minutes, and there are different types of rapid tests. These are very easy to perform and do not require any special instrument. So like ELISA, we do not require any special instrument here. These examples of these rapid test is dot blot assay, or for example is tri-dot test, which is commonly done for diagnosis of HIV. It can detect HIV1 and 2 separately. This is in a cassette form. This is a cassette. It consists of a nitrocellulose membrane and an absorbent pad. Nitrocellulose membrane is coated at 3 regions. 2 test regions, you can see HIV1 and HIV2, and above one is a control region. These test regions, HIV1 and HIV2, these are coated with HIV1 and 2 antigen, and the control region is coated with anti-human gamma globulin. Now we will see how the testing is done. As I have told you, first the sample is collected in these test tubes. Now for sample collection, you have to remember 3 C's. Now coming to the test, we will see how it is performed. This is a tri-dot test, which is a dot blot assay. Now first the sample ID is put on the cassette. Then first we will have to add buffer solution, 2 drops. It contains absorbent pad, so it will absorb the additional amount. Then once it is done, then sample is taken and it is added to this cassette. You will have to use gloves you will have to take the standard precautions while doing any type of blood test or whenever you touch blood or body fluids. You will have to perform with standard precautions. Then again buffer is added after adding the sample. Buffer is added 3 drops. Then once it is absorbed, we will have to add the conjugate. Conjugate contains the enzyme which will give a pinkish purple color. Now see the result. Let the conjugate get absorbed to the membrane. Now can you see at the top there is a control dot. That is a control dot. That means you have performed the test. Then to the left you can see HIV-1 is written and it shows a dot also. So that means this sample is positive for HIV-1. And to another side there is no dot, that means it is negative for HIV-2. This is how you perform the HIV testing. Now after this you will have to discard these samples as well as this cassette properly, taking all the precautions, considering the rules of biomedical waste management. So this is a simple rapid test and you have seen the result within minutes. Thank you.